Oh no, you can see it. Never mind. Oh my god, I couldn't see it. Sorry. Now there's a GLUT2 transporter here, which is the fourth and final carrier in the intestinal absorption of glucose, galactose, and fructose, and it works for all three glucose monosaccharide carbohydrate monosaccharides, glucose, galactose, and fructose. And that's how it's going to leave out of the cell, since there's high concentrations of all three here and low concentrations on the blood vessel side. It can easily just go out by facilitated diffusion with the help of the GLUT2 transporter. And here I wrote a note for you guys that absorption of monosaccharides does not take place in the colon, as there is no transporters. So this mainly happens in the small intestine, okay? Now, fermentation in the colon. As I just said, there is no absorption of monosaccharides in the colon, right? So what happens to the glucose, the galactose, the fructose molecules that are found in the colon? Fermentation. And that is because of those 4-5 criterias that help in the fermentation taking place in the colon. First of all, fermentation is an anaerobic process. And I can't type at all on this. Well, I'm sorry. I hope it's clear. Okay? It's an anaerobic process, which means it occurs when there is low oxygen or no oxygen at all, which is here because partial pressure of oxygen decreases along the GIT tract, which means by the time you reach the colon, there is almost zero PO2. Well, not zero, but very low PO2 concentrations. Colon has sluggish motility except during mass movements, which means that the micronutrients or the monomers, the glucose, galactose, fructose molecules, they stay in the colon for a really long amount of time. So the bacteria has a lot of time to feed on those monomers. So it's not like in the small intestine. The small intestine has a lot of motility. There's the MMC, there's the peristaltic contractions every 9 to 12 minutes, the emptying right after eating, the strong antral peristalsis. So in the upper GIT tract, there isn't really a lot of time for anaerobic fermentation to take place. As well in the upper part of the GIT, there are bile salts which disrupt bacterial cell membrane. So bacteria doesn't even have the correct adaptations for it to be able to live in the small intestine and the upper GIT tract, as well as the acidic environment in the stomach, as you know, kills the bacteria. However, in the colon, there is it's high in pH, low PO2. It doesn't move. The colon doesn't really move. It has mass movements, which happens every hours, like a couple of hours, like 8 or something. I'm not really sure. And little bile salts and no pancreatic enzymes, which keeps it as a basic environment for the bacteria to be able to live in. And this is why you see here, where does fermentation start to happen? In the ileum. This is the number of organisms that are found in the stomach, duodenum, jejunum, ileum, and the large bowel. Of course, the large bowel has the highest amount of organisms because it has the ideal environment for the bacteria to live. However, the stomach, the duodenum, and the jejunum, kind of the ileum. The ileum is where there is less acidic environment, there is less PO2, there is less motility. So this is where the bacteria start to be able to adapt to that environment and grow. So here you'd see that the log 10 of the organisms, just ignore that. You just need to understand that this is the number of organisms in the stomach, duodenum, in the GIT basically. It's around log 2 or like this, 2 point something. However, in the large bowel, it's almost 12. Like it's really high, like 11.5 or something. Now, the carbohydrate fermentation by colonic bacteria, cellulose. Cellulose is there is no enzyme in the human body that can break down cellulose. So how is cellulose broken down or digested? It's digested by fermentation, basically. And the products would be short chain fatty acids such as acidic propionic butyric acid, which are all absorbed by the colon by short chain fatty acid, sodium co-transported. So instead of as we've seen before, was the asymptoter or the co-transport of sodium with glucose and galactose, which was the CGLT1 LT1, okay, that would absorb the sodium and galactose and glucose into the enterocyte by the taking in of sodium. We have the same concept, but for short chain fatty acids in the colonocytes, so way down in the GI tract in the colon. And gases are the second product. So we have short chain fatty acids and gas. What type of gases do we have? We have carbon dioxide, methane, and hydrogen. And those can be absorbed into the blood or expelled through the anus. Now, osmotic diarrhea in the case of lactase deficiency. We said that lactase is the enzyme that breaks down lactose into galactose and glucose in the brush border, right? So if you lack this enzyme, of course, there is no other way for lactose to be broken down. So how is it going to be broken down? It's going to be broken down by fermentation down in the colon, and my handwriting is really bad. I apologize. So it's going to be broken down by bacteria. 2 carbon metabolites such as acetic acid, which are the short chain fatty acids that we were talking about. Fatty acids. You're gonna have 3 carbon metabolites, lactic acid, CO2, and H2. So those are found in the small intestine, right? So let's say this is the lumen of the small intestine, which is where this is right now. I mean large intestine, I'm really sorry. This is the large intestine because we're talking about fermentation. So this is the lumen of the large intestine. Now we have a lot of metabolites. So the solute molecules, the solutes are a lot higher now. The concentration is a lot more. While the interstitium or the blood, which is around, let's say here, it's going to be a lot lower. So where is the movement of water going to be? From low to high. So the movement of water is going to be from low to high, increasing the water concentration in the large intestine, which causes bloating and diarrhea, which is why people that don't have the lactase enzyme frequently have bloating and diarrhea. In extreme cases of lactose intolerance, usually fiber-rich bulking agents are commercially available to stop bloating and diarrhea. How do you test for lactase deficiency? First, you give patient lactose, only lactose. So you won't give him glucose, galactose, any of those, so that you are solely focusing on lactose and make sure that the lactase enzyme is what you're testing for, whether or not the patient has lactase deficiency. And then you measure the hydrogen concentration in the patient's breath after 2 hours, because it usually takes 2 hours for the lactose to reach the large intestine or the small intestine where lactase is, and large intestine for fermentation. This is how the device looks like. The one you measure the hydrogen concentration with in the patient's breath, obviously. And this graph, one second. There we go. Now this graph shows the hydrogen and the amount of hydrogen in the breath test over the amount of time. So as you can see, after 2 hours, when it increased, look, it dramatically increased. The hydrogen concentration dramatically increased, which tells us that this patient is lactase deficient. Now, there's another deficiency, which is the Sucrase-isomaltase deficiency, the complex, which are like they're one complex, the one that looked like this when I drew it, something like this, I think. Okay, in the brush border of the intestine. This is a congenital defect. Patients usually have sucrose and starch intolerance. Sucrose because obviously the sucrase enzyme is deficient. And why starch? Starch because 75% of starch, as we said, is amylopectin. And amylopectin gives us alpha amylase, alpha limit dextrin. Alpha limit dextrin, which is broken down by isomaltase, because isomaltase is the only enzyme that breaks down alpha-1,6 glycosidic bonds. Now, this is a brush border complex, as I've said before. Pediatrics starts after weaned off milk and starts what? It usually starts in kids. After you've weaned off the milk and started juices, symptoms are the same as lactose intolerance. And you can also test sucrose as a substrate of breath hydrogen. I just need to fix this. I'll fix it before we upload the lecture. Now, carbohydrate malabsorption pathogenesis of the symptoms. Now, when you malabsorb dietary carbohydrate, which means you can't absorb the carbohydrate, so whether it's lactose, glucose, sucrose, any of those, you'll either have an osmotic load, as I've said before, for example, in the lactose example. You've had an increase in solute molecules, which causes the water movement from the interstitium into the lumen, which this is called an osmotic load. And then you have the increase in fermentable substrate, because of course if it reaches into the colon, you don't have transporters for those substances in the colon, so they get fermented, which increases gas production, because one of the products of fermentation is carbon dioxide, methane, or hydrogen gas. This causes the osmotic load, if it increases luminal fluid, which causes luminal distension, or bloating, which causes bloating, diarrhea, pain, and gas. Now, hydrolysis of ingested nutrients. Now we're talking about proteins. For the carbohydrates, were able to be digested in the lumen, the microvilli, but not in the enterocyte, so not in the cytoplasm of the enterocyte. This is supposed to be cytoplasm, my bad. Okay. However, proteins are digested in all three places. Proteins is a dietary source. The dietary sources of proteins is in meat, eggs, and soybean, quinoa, all of those, even chicken. It's an endogenous source. Enzymes, mucoproteins, etc. You can those are from secretions. Those are made up of proteins, but they're secreted. We make them in our body, and they are shed by epithelial cells. Now, protein digestion, luminal hydrolysis. So in the lumen, we are talking about here right now. We're here. Two locations. You either digest protein in the gastric lumen or the intestinal lumen. In the gastric lumen, which is in the stomach, pepsinogen is released. Pepsinogen is then with increase of hydrochloric acid release from chief cells. It activates pepsinogen to pepsin. This is at around a pH optimum of 2 to 3. So optimum pH for pepsin is here between 2 to 3. And then in the intestinal lumen, inactive pancreatic enzymes are released and are then activated by enteropeptidases. So the pancreas secretes enzymes, and then enteropeptidases are released to activate those inactive pancreatic enzymes. So yeah, and the pH optimum is 6 to 8. Side note, secretion of pancreatic enzymes are enhanced by cholecystokinin. So release of cholecystokinin when you eat food, it pressures on the ampulla of the pancreas to secrete more pancreatic enzymes for protein digestion. So there are two types of enzymes for protein digestion. There's either endopeptidases or exopeptidases. So the endopeptidases, if this is a peptide polymer, the ones on the end, the last ones, are broken down by exopeptidases, while the ones in the middle are broken down by endopeptidases. So the endopeptidases, they digest the internal peptide bonds, example pepsin in the stomach, trypsin and chymotrypsin in the small intestine, while exopeptidases, they digest the terminal bonds, so the last ones on the edges, to release amino acid, which is found in two types as well. They're either aminopeptidases, which are not found in humans, or carboxypeptidases, which we have in two forms, carboxypeptidase A and carboxypeptidase B. Let me just delete the screen. So as we said, proteins, you ingest it, and then in the stomach, pepsinogen is converted into pepsin by H+, which is taken from hydrochloric acid, which is released by chief cells, and pepsin is an endopeptidase. So this is one of the endopeptidases which break down the internal bonds. This is in the gastric lumen, which gives us all of those. Native protein, protein fragments, large polypeptides, and small amounts of free amino acids. So like you won't find a large amount of amino acids broken down by endopeptidases because they break down the internal bonds and not the terminal bonds. And then when those proteins move into the intestinal lumen, they are broken down further to di and tripeptides, peptides or amino acids, but mostly di and tripeptides by either trypsin, chymotrypsin, which are endopeptidases, so they'll break down the internal bonds, or carboxypeptidases A and B, which are exopeptidases, and those are the ones that usually give out the free amino acids. Now we've done the lumen. We're moving on to the protein digestion of in the brush border and intracellular hydrolysis. So the brush border peptidases will hydrolyze any remaining oligopeptidases to di and tripeptidases and amino acids. Brush border peptidases will hydrolyze any remaining oligopeptides to di and tripeptides and amino acids. After hydrolysis on the brush border, there are transporters that will take in those di and tripeptides into the enterocyte. However, peptides cannot be taken in, so either amino acids or di and tripeptides will be able to be taken in to the enterocytes. And then inside the cell, the di and tripeptides are further broken down to amino acids by cytoplasmic peptidases. Now, the peptide transport. The first one is the exact same mechanism as when we were talking about carbohydrate absorption. Sodium 3, you use the sodium potassium ATPase pump to take out 3 sodium molecules from inside the cell, taking in 2 potassium molecules using ATP. This will allow the second sodium hydrogen exchanger to work, taking in sodium and taking out hydrogen. This will further allow the peptide di and tripeptides to work by the PEPT1 carrier, the hydrogen peptide co-transport PEPT1. Okay, so the hydrogen which was taken out from using the sodium hydrogen exchanger will be taken in again by the PEPT1, with it the dipeptide and tripeptide. So this is a symptoter, and it has broad specificity, so it won't, like, any type of di and tripeptides can be taken in. And then we have the intracellular peptidases, which hydrolyze the di and tripeptides into free amino acids, and then the free amino acids will leave the cell by facilitated diffusion using a carrier and into the bloodstream. This is just this is to remind you of these carrier system of the carbohydrate mechanism. Sodium out, potassium in. Using ATP, we will take in sodium as well as glucose. Now, digestion of ingested proteins, we've moved on to the lipids. So lipids can only be broken down in the lumen, so it cannot be broken down in the microvilli or the enterocyte, which is in the cytoplasm, like the cytoplasm of the enterocyte. However, they require pancreatic enzymes and